and welcome. My name is Kateri Scott, and thank you for joining me today for this presentation on moss magic, harnessing photosynthesis for air purification. Meet the tiniest environmental superhero you've probably never noticed, moss. While trees get all the attention for cleaning our air, these little green cushions might be even more effective in purifying our air than you might think. The unseen environmental heroes. As we begin our exploration, I'd like to introduce you to what I call the unseen environmental heroes of our planet. Mosses are remarkable organisms with ancient origins dating back over 450 million years. These plants evolved from aquatic environments to become some of the first colonizers of land, developing specialized adaptations that allow them to thrive in diverse environments. What makes mosses particularly fascinating is their non-vascular design. Unlike most plants you're familiar with, mosses lack specialized transport tissues like xylem and phloem, instead absorbing water and nutrients directly back through their leaves. This unique characteristic, as we'll see later, gives them exceptional capabilities for air purification. Their ecological contributions are significant and multifaceted, from stabilizing soil through their dense growth patterns to sequestering carbon through photosynthesis. They excel at water retention in ecosystems, and most relevant to our discussion today, air purification through bioaccumulation of pollutants. Ancient origins and evolutionary journey. To appreciate moss fully, we need to understand their evolutionary journey. Their story begins over 450 million years ago. The aquatic green algae known as charophytes during the Ordovician period, as land began to be colonized by plants, early moss ancestors developed crucial adaptations for surviving in terrestrial environments. The transition from water to land was no small feat. These pioneering plants needed to develop mechanisms for desiccation tolerance and nutrient acquisition from soil rather than water. Throughout the Devonian period, we see the clear emergence of birophytes, the plant group that includes moss. And by the Carboniferous period, they had diversified and colonized various terrestrial niches. Key evolutionary adaptations that enabled their success include remarkable desiccation tolerance, structural support systems that allow them to grow in air, specialized reproductive structures adapted for terrestrial dispersal, and their distinctive non-vascular tissues for nutrient transport. These adaptations were responses to environmental pressures such as UV radiation, desiccation risk, nutrient limitation, and competition. The life cycle of moss is quite different from that of flowering plants most people are familiar with. Moss have what botanists like to call gametophyte dominant life cycle, where the main visible plant, the green leafy structure we recognize as moss, is actually the haploid gametophyte generation. This gametophyte produces male and female reproductive structures called antheridia and archegonia. Sorry, those are some difficult words. Okay, when water is present, sperm with the antheridia swim to the archegonia, where fertilization occurs. This creates a zygote that develops into the sporophyte generation, the often brownish capsule-like structure you might see rising from a moss mat on a thin stalk. The sporophyte remains attached to and nutritionally dependent on the gametophyte. Inside the sporophyte capsule, meiosis occurs to produce haploid spores. When conditions are right, these tiny spores are released and dispersed by wind or water to new locations, where they germinate into a filamentous structure called a protonemia. This eventually develops into a new gametophyte plant, completing the cycle. This unique reproductive strategy allows moss to colonize new habitats efficiently and maintain genetic diversity, contributing to their remarkable resilience and widespread distribution. In ecological succession, moss often serve as pioneer species, the first organisms to colonize disturbed or barren environments. Their role in this process is multifaceted and crucial for ecosystem development. First, moss excel at soil stabilization. Their dense growth forms protective mats that prevent erosion on exposed soil surfaces. As they grow, they trap soil particles and organic matter, gradually building a more hospitable substrate for other plants. Water retention is another vital ecological function. Moss can absorb and hold many times their weight in water, creating moisture reservoirs in otherwise dry environments. This moisture retention moderates local humidity and provides water access for other organisms. Moss also create microhabitats that support diverse communities of microorganisms, insects, and other small invertebrates. The sheltered, moist environments they create serve as nurseries for these organisms, enhancing local biodiversity. Finally, by modifying their physical environment and facilitating the establishment of other organisms, moss advance ecosystem succession, paving the way for larger, more complex plant communities and the animals that depend on them. This ecological foundation building makes them crucial components of healthy, resilient ecosystems. The remarkable air purification capabilities of moss stem largely from their unique anatomical structure. Unlike vascular plants, moss have an exceedingly simple body plan, with leaves that are typically only one cell thick. This minimal barrier between internal cells and the external environment allows for direct gas exchange and absorption across the entire plant surface. The stem-like structures in moss are also quite simple, often lacking specialized conducting tissues. Instead, they primarily provide structural support, with water and nutrients moving through the moss mainly via capillary action and diffusion. At their base, moss have thread-like structures called rhizoids rather than true roots. These rhizoids anchor the plant to its substrate, but don't play a significant role in water and nutrient absorption as roots do in vascular plants. This simplified anatomy creates an enormous surface to volume ratio, maximizing the area available for environmental interaction. Essentially, the entire moss plant becomes an interface for gas exchange and pollutant absorption, a living filter with every cell potentially participating in air purification processes. The non-vascular nature of moss, which might seem like a limitation, is actually a significant advantage for air purification applications. Without specialized vascular tissues like xylem and phloem, moss have evolved to absorb water nutrients and, by extension, pollutants directly through their surfaces. This direct absorption happens across the entire plant body, with each cell potentially in contact with the environment. Water and dissolved substances move through capillary action between overlapping leaves and through specialized water-conducting cells called hydroids in some moss species. This mechanism allows moss to efficiently extract substances from their surrounding environment, including airborne pollutants. Particulate matter and gaseous pollutants that come in contact with the moss surface can be trapped, absorbed, and in some cases metabolized by the plant. The absence of a cuticle, the waxy protective layer found on most vascular plants, further enhances this absorption capacity. Without this barrier, gases and water-soluble pollutants can pass more freely into moss tissues, making them particularly effective in biological monitors and remediators of air pollution. At the heart of moss's air purification capability is photosynthesis. The process by which plants convert light energy into chemical energy. This slide illustrates a simplified overview of this remarkable process in moss. Starting on the top left, we see the chlorophyll pigments in the thylakoid membranes of the moss cells. They capture light energy from the sun. This energy drives the conversion of carbon dioxide and water into glucose and oxygen. As shown in the bottom process flow, unlike most vascular plants that regulate gas exchange through specialized stomata, mosses absorb carbon dioxide through their entire leaf surface via simple diffusion. This is possible because moss leaves are typically only one cell thick, allowing gases to freely diffuse in and out of their tissues. As photosynthesis proceeds, oxygen is released as a byproduct, a process that not only supports the moss's metabolism but also refreshes the surrounding air. This oxygen production, coupled with carbon dioxide consumption, represents the most basic way mosses contribute to air quality. Moths have several adaptations that enhance their photosynthetic efficiency and their particular ecological niches. Their thin leaves allow for efficient gas diffusion, and the absence of stomata in the gametophyte stage means they engage in direct surface exchange with their environment, maximizing their interaction with atmospheric gases and pollutants. Diving deeper into the photosynthesis process, let's examine the Calvin cycle. This should look a little familiar. The Calvin cycle, the series of chemical reactions that actually fix atmospheric carbon dioxide into organic compounds. The Calvin cycle consists of 3 phases. First, in carbon fixation, atmospheric CO2 is combined with a 5-carbon sugar called ribulose 1,5-biphosphate, or RUBP. Through the action of an enzyme called rubisco, this creates an unstable 6-carbon compound that immediately breaks down into 2 molecules of 3-PGA. Second, in the reduction phase, ATP and NADPH energy carriers produced during the light-dependent reactions we discussed earlier are used to convert the 3-PGA into the G3P, a 3-carbon sugar. Finally, in regeneration phase, some G3P molecules are used to regenerate the RUBP that started the cycle, ensuring the process can continue as long as light energy is available. For 3 cycles, 1 extra G3P molecule becomes available for the plant to use in synthesizing glucose, cellulose, and other organic compounds. This fixed carbon becomes part of the plant's biomass, and importantly for our discussion on air purification, represents carbon that has been removed from the atmosphere and sequestered in the moss tissue. The efficiency of the cycle in moss, combined with their extensive surface area for gas exchange, makes them particularly effective carbon captors relative to their size. Moving on from internal processes to external interactions. Moss employ several physical mechanisms to filter particulate matter from the air. The dense growth habit of moss creates intricate mats with numerous tiny spaces between individual plants and leaves. This 3-dimensional structure functions similarly to engineered air filters, creating a maze that airborne particles must navigate. As air flows through this natural labyrinth, particles collide with moss surfaces and become trapped. The high surface area to volume ratio of moss gametophytes maximizes the potential for particle interception. Each tiny leaf and stem presents another opportunity for particulate matter to be captured from the passing air. Many moss species also secrete mucilaginous compounds that coat their surfaces, creating sticky traps for airborne particles. Once captured in this adhesive coating, particles are effectively removed from circulation and can eventually be washed into the substrate by rainfall or incorporated into the moss tissue itself. Research has shown that urban moss mats can trap significant amounts of particulate matter, including harmful PM10 and PM2.5 particles that pose serious health risks when inhaled. This physical filtration occurs passively and continuously, making mosses valuable allies in combating particulate air pollution. Beyond trapping particles, moss are also adept at absorbing chemical pollutants from the air. Their high surface area and lack of protective cuticle allows for efficient uptake of gaseous pollutants through the entire plant surface. Volatile organic compounds, or VOCs, chemicals emitted from many household products, paints, and industrial processes, can be absorbed by moss tissues and in some cases metabolized or sequestered. Studies have shown that certain moss species can reduce indoor VOC concentrations by significant percentages when incorporated into living wall systems. Nitrogen oxides from vehicle emissions or other combustion processes are also effectively captured by moss. The plant tissues can absorb these compounds directly from the air, potentially converting them to less harmful forms through the various metabolic pathways. Similarly, sulfur dioxide, a major component of acid rain and industrial pollution, is readily absorbed by moss tissues. While high concentrations can damage the moss itself, moderate levels can be effectively removed from the air and and incorporated into plant biomass. This chemical absorption capacity makes moss valuable tools for monitoring and mitigating air pollution, particularly in urban environments where these pollutants are most concentrated. Once pollutants are captured or absorbed, moss employs several strategies to manage these potentially harmful substances. For many organic compounds, moss possess enzymatic systems that can break down the pollutants into less harmful components. These detoxification mechanisms vary by moss species and pollutant type, but can include processes similar to those found in the liver of humans, such as oxidation, reduction, or conjugation reactions that render toxins less harmful. Heavy metals present are a different challenge, as they cannot be broken down. Instead, moss excel at sequestering these metals, binding them to the cell wall components or storing them in vacuoles or specialized proteins where they cannot interfere with the cellular process. The cell walls of moss contain compounds like polyglutaronic acid and have a high affinity for metal ions, effectively immobilizing them. This bioaccumulation capability is also pronounced that mosses are widely used in environmental monitoring as bioindicators of heavy metal pollution. Scientists can analyze moss tissues to assess environmental exposure levels, as the moss effectively samples the air over time, accumulating pollutants that might be present at levels too low for conventional monitoring equipment to detect consistently. While this bioaccumulation is beneficial for removing pollutants from the air, it does mean that heavily polluted moss may eventually need replacement in engineered systems, as its capacity for additional uptake may become saturated. Among moss species, sphagnum moss deserves special attention for its extraordinary carbon sequestration capabilities, often called peat moss. Sphagnum is the primary component in the formation of peat bogs, ecosystems that, despite covering only about 3% of the Earth's land surface, store approximately 30% of all soil carbon. This remarkable carbon storage capacity stems from several unique properties of sphagnum moss. First, its tissues are highly resistant to decomposition due to phenolic compounds, and other substances that inhibit microbial activity. When sphagnum dies, rather than quickly decomposing, it accumulates as peat. Additionally, sphagnum creates acidic waterlogged conditions in its environment through cation exchange processes, further slowing decomposition. The moss releases hydrogen ions and absorbs calcium, magnesium, and other cations, progressively acidifying its surroundings. Its exceptional water retention capacity, sphagnum can hold up to 20 times its dry weight in water. This creates anaerobic conditions that prevent complete decomposition of organic matter. This combination of chemical inhibition and physical environment manipulation results in carbon being locked away for thousands of years rather than returning to the atmosphere. From a climate perspective, preserving existing peat bogs and potentially creating engineered systems that mimic these natural carbon sinks represents a valuable strategy for long-term carbon management. Moving from natural ecosystems to human applications, let's explore how moss can be integrated into indoor environments for air purification. Moss terrariums provide a compact solution for smaller spaces. These self-contained ecosystems not only serve as attractive decorative elements, but also function as natural air purifiers. The enclosed or semi-enclosed environment allows moss to trap particulate matter and absorb volatile organic compounds that are common in indoor air, such as formaldehyde from furniture and carpets or benzene from electronic devices. On a larger scale, living moss walls represent a more ambitious application. These vertical installations feature growing moss on a substrate that can be integrated into interior architecture. Beyond their striking visual appeal, moss walls provide substantial air purifying surface area. Research has shown that moss walls can significantly reduce indoor CO2 concentrations, capture airborne dust, and absorb various VOCs. When strategically placed in high-traffic areas or spaces with limited ventilation, these living systems continuously work to refresh the air. Unlike mechanical air purifiers, they operate silently, consume no electricity, and add humidity to often dry indoor environments, a benefit for both human comfort and preventing airborne disease transmission. The psychological benefits of bringing these natural elements indoors should not be overlooked either. Multiple studies have documented improved mood, concentration, and productivity in spaces incorporating living plants, adding another dimension of value to these moss applications. Expanding to the urban scale, moss installations offer promising solutions for city-level air quality challenges. Green roofs incorporating moss species provide multiple environmental benefits. They absorb pollutants directly from urban air while also managing stormwater runoff that would otherwise carry pollutants into waterways. The natural insulation properties of moss layers contribute to energy efficiency by reducing building heating and cooling needs. Perhaps most importantly in urban settings, moss-covered surfaces help mitigate the urban heat island effect through evapotranspiration, releasing moisture that cools the surrounding air. Vertical gardens, or living walls, featuring moss, serve as large-scale biological air filters in high-density urban areas. These installations can be integrated into building facades, highway sound barriers, or public infrastructure, creating multifunctional surfaces that combine aesthetic value with air purification. Moss are particularly well suited for vertical applications due to their lightweight nature and minimal soil requirements compared to other plants. The heat island mitigation aspect deserves special emphasis. Urban areas typically experience temperatures 1 to 7 degrees Celsius higher than surrounding rural areas due to heat absorbing surfaces and human activity. Moss installations help counteract this effect through evapotranspiration. This process by which moisture is released into the air, creating natural cooling. A network of moss installations throughout a city can contribute to meaningful temperature reductions while simultaneously filtering air pollutants. To illustrate the practical application of moss-based air purification, let's examine the city tree technology, one of the most successful implementations of engineering moss biofilters for urban settings, developed by Green City Solutions. City trees are freestanding installations that combine moss cultures with smart technology to create high-efficient air filtration units. Each unit contains the air cleaning capacity equivalent to up to 275 actual trees, but requires just 1% of the space and 5% of the cost. The system's effectiveness stems from optimized moss species selection and an integrated integrated IoT Internet of Things monitoring system. Sensors continuously track air quality parameters around the installation, while automated irrigation and climate control ensure optimal growing conditions for the moss, maximizing its air purification performance. Field tests in major European cities have demonstrated significant local air quality improvements, with reduction in nitrogen oxides and particulate matter of up to 53 and 30% respectively, within the immediate vicinity of the installations. The units are particularly effective when placed at pollution hotspots such as busy intersections or bus stops. The real-time monitoring capabilities not only optimize the performance of each unit, but also generate valuable data on urban air quality patterns that can inform broader environmental planning. Multiple cities, including Berlin, Oslo, and London, have implemented these units, providing real-world validation of moss-based air purification technologies at an urban scale. Despite their resilience, successful moss-based air purification systems must address several environmental requirements and challenges. Moisture needs represent the primary consideration. Most moss species require consistently damp conditions to maintain optimal metabolic activity. and thus their air purifying functions. In natural settings, morning dew or regular rainfall might suffice, but engineered systems typically require irrigation, infrastructure, particularly in dry climates or indoor applications. Many moss species also prefer shaded or partially shaded conditions. Direct, intense sunlight can lead to desiccation or reduced performance, or even damage to the moss tissues. This light sensitivity must be accounted for in placement and design of moss installations. Humidity levels are closely related to moisture needs but deserve separate attention. Most moss thrive in environments with relative humidity above 50%, which can be challenging to maintain in air-conditioned indoor spaces or in naturally arid regions. The greatest vulnerability for moss systems is desiccation, the drying out of tissues during extended dry periods. Without adaptations or technological support, moss installations can become dormant or even die when subjected to prolonged drought conditions. While many moss species can recover from desiccation when moisture returns, their air purification functions are significantly reduced during dormant periods. These challenges are not insurmountable, but require thoughtful system design, species selection, and potentially technological support for automated misting systems or humidity control in indoor applications. Fortunately, not all moss species are equally vulnerable to environmental stresses. Through careful species selection and leveraging natural adaptations, many of these challenges we discussed can be addressed. The sphagnum species, which we discussed earlier for their carbon sequestration abilities, also demonstrate exceptional water retention capabilities. Their unique cell structure includes specialized dead cells called hyaline cells that can hold water even in relatively dry conditions, maintaining humidity around the living cells. Birum species are known for their resilience in variable environments, including urban settings with fluctuating moisture levels. Their compact growth, form, and ability to recover rapidly from desiccation make them suitable candidates for engineering air purification systems. Several moss species have evolved remarkable desiccation tolerance strategies. When water becomes scarce, they can enter a state of suspended animation, essentially shutting down metabolic processes until conditions improve. Upon rehydration, they can resume normal function within hours, a feat few other plants can match. This resilience can be further enhanced through various engineering approaches. Substrate selection affects water retention and availability to the moss. Capillary irrigation systems can deliver precise amounts of water directly to the moss, and for truly challenging environments, climate-controlled enclosures can maintain optimal conditions regardless of external circumstances. By matching species to application conditions and incorporating appropriate support systems, moss-based air purification can be made viable across a wide range of environments. The field of moss-based air purification holds tremendous potential for innovation and advancement. Current research is focusing on several promising areas. Bioengineering efforts are exploring how to enhance the natural capabilities of moss through selection and potentially genetic modifications. By identifying and propagating moss variants with superior pollutant absorption rates or greater environmental resilience, researchers hope to develop supermoss, specifically optimized for air purification applications. Optimizing large-scale carbon capture represents another active research direction. Building on our understanding of how sphagnum moss create carbon sequestering peat bogs, scientists are investigating ways to accelerate and maximize carbon storage in engineered moss systems. This could potentially create carbon negative installations that not only filter air pollutants but also help mitigate climate change. Hybridization with other technologies show particular promise for expanding the effectiveness of moss-based systems. Combinations with traditional mechanical filtration, photocatalytic materials, or even bioengineered bacteria can create multi-layered purification systems that address a wider range of pollutants than moss alone. Monitoring and quantification methodologies are also advancing rapidly. More sophisticated sensors and analytical techniques allow for precise measurement of moss system performance, generating data that can further refine design parameters, and demonstrate effectiveness to potential adopters. These research directions suggest that we've only begun to tap into the potential of moss-based solutions for environmental challenges. As our understanding grows, as too will the sophistication and effectiveness of these applications. Looking toward the future, moss-based technologies have the potential to become integral components of sustainable urban design. Integrated city planning could incorporate moss installations as standard elements in urban infrastructure, from highway sound barriers and building facades to public transit shelters and street furniture. This distributed approach would create a network of biological air filters throughout the urban landscape, working continuously to improve air quality. Smart city integration represents the next evolutionary step, with moss installations connected to urban monitoring networks. These systems could track air quality in real time, adjusting irrigation or ventilation parameters based on current pollution levels or weather conditions. Data from these networks could inform public health alerts or traffic management during high pollution events. These technologies contribute to the broader concept of biomimetic urban design, the practice of emulating natural systems to solve human challenges. By incorporating nature's time-tested solutions like moss-based air purification into our built environment, we create more resilient, livable cities that work with natural processes rather than against them. The holistic environmental benefits extend beyond air quality. Moss installations contribute to urban biodiversity, stormwater management, noise reduction, and aesthetic improvement. These combined effects support a systems-based approach to urban environmental challenges, where a single intervention delivers multiple benefits across different domains. As we continue our exploration of moss-based air purification, let's synthesize what we've learned. The biological mechanisms we've examined: photosynthesis, cellular respiration, bioaccumulation, and carbon sequestration work together as an integrated system that improves air quality on multiple fronts. What makes these mechanisms particularly valuable is that they've been refined through millions of years of evolution, resulting in high efficient processes that operate as ambient temperatures and pressures without harsh chemicals or energy inputs. We've seen how moss support broader ecosystem stability while providing natural air purification services. By incorporating these organisms into our outdoor and urban environments, we're not merely adding decorative elements but functional biological systems that continuously work to clean the air around us. The elegance of moss-based solutions lies in the connection they represent, a bridge between ancient biology and innovative applications that address contemporary challenges. By understanding and applying these natural mechanisms at scale, we can develop sustainable solutions that contribute to healthier living spaces and more resilient urban environments. As we face growing environmental challenges, particularly in our own densely populated urban areas, perhaps the most powerful solutions will come not from inventing entirely new technologies, but from better understanding and applying the sophisticated biological systems that nature has already perfected. This final slide presents our bibliography and references. The information shared today draws from a range of authoritative sources, including academic textbooks like Clark's, peer-reviewed research papers such as Chidori and Roy's systematic review on moss-based air quality monitoring, and reports from organizations like Cordis on urban air quality improvements using moss-based filters. I'd like to acknowledge that AI assistance tools were used in the brainstorming and drafting process for this presentation, with all information verified against these peer-reviewed sources to ensure accuracy and scientific validity. All references are formatted according to APA style, and the complete citations are available for anyone interested in exploring these topics further. Thank you for your attention. I hope you enjoyed my presentation on the fascinating world of moss and its applications for air purification.